This is part C. There are many aspects that would go into isolating a gene from a eukaryote to express in a prokaryote. This is referred to as lateral gene transfer, which is the transfer of genes not involving direct vertical inheritance. Gene transfer is not common or natural, especially for the transfer between a eukaryote to a prokaryote, so there are many requirements for this event to occur. First, you need to know the sequences of the transferred gene and the bacterial genome. You know this because there are many differences between the gene expression in prokaryotes versus eukaryotes. For example, in eukaryotes, one mRNA is equal to one protein. However, in bacteria, one mRNA can code for multiple proteins. The reason you would need to know the sequencing is because genes are much more organized and repetitive in eukaryotes because they're structured together in histones. But only about 2% of the entire genome codes for proteins in eukaryotes. But in bacteria, around 95% of the genes will code for proteins, and they're not split by exons and introns, which make it even harder to identify their location within the genome. Another requirement is that eukaryotic genes must be highly conserved, which means that the genes must be relatively unchanged throughout evolution. This allows there to be a possible common ancestor to assist the transfer. In addition to gene conservation, it is a requirement that the protein must have little variability. If it is flexible, then it can be catalyzed by different enzyme forms with evolutionary histories that line up with the transferred gene and the bacteria. I was able to make changes to the eukaryotic gene sequence before inserting it into the bacteria. I would have to know the protein sequences of both the eukaryote and the prokaryote DNA. Then I would have to align them according to their base pairs. If the protein sequences are rearranged, I would need an enzyme that is able to reconstruct the relationship between the prokaryote genes and the eukaryotic genes in order to make a functional prokaryote.